attendance. And so thank you all. And to those who are online, I hope you're all safe, and I hope you can join us someday in the future. We hope the transmission is working well. I'm sure it is. But we have an interesting program for you tonight. It's certainly different from the one that we had last month when we had our lovely Susan B. Anthony. Not that you're not lovely, Jim, okay? So, but the program is not what I expected when I was first given the title 6 months ago. A humongous fungus among us. I thought it was about Washington. But anyway, on the speaker tonight is our own Jim Coppinger. And those of us on the board think of him as Mr. Kalamazoo Torch. He makes the whole thing work every month. Thank you, Jim. Appreciate it. Well, since you all know who Jim is, then I don't need to do a long introduction. I'll just read the bio that he sent me and sit down. Jim's a graduate of Western with a bachelor's in printing management and business. He began his advertising career as an advertising sales rep for the Gazette, where he later became marketing director and then advertising director for the paper. In 2001, he started Quadrant Marketing, a boutique advertising, marketing, and PR agency. Jim is also engaged in professional stock photography and video. He's had a lifelong interest in science, particularly the natural sciences, and spends much time in the outdoors. He and his wife, Joan, have 3 grown children and 5 grandchildren. Okay, Jim, the floor is yours.
All right, am I live? Well, maybe I could preface a little bit that, when they talk about people, public speaking is just a terrifying event. Tonight, for me, is going to be just absolutely a piece of cake, because as I was telling Dave and Connie, for some unknown reason, probably about 4 weeks or so ago, I had a dream, and the dream was I was here to give this presentation, and probably sitting right here, pretty close to this, having conversation, and just finishing up the dinner so it was ready to start, and I realized that I'd brought everything else but this, my program, and so I said to someone, tell Jeff I'm just going to run home because I'm not that far from here. I'll run home, I'll get the notebook and shoot back, so just keep it going, but I'll be right back. And when I came back, because of all the construction with the park club, you couldn't get in, we couldn't find an entrance, and then some woman said, well, I know how to get you to that room, and we're wandering all these bizarre rooms. I don't think I ever gave that presentation, so again, this is going to be easy. So yes, there is a humongous fungus that lives among us alongside the world's largest radio station in Michigan's Upper Peninsula. This story really begins deep in the oceans with the elusive naval submarines that carry out months-long missions. And while the world's largest animal, the blue whale, lives below the surface of the ocean, the largest organism on Earth also lives below the surface, but on land. So tonight I will share the story of the discovery of a fungus, Armillaria gallica, the people who discovered it and continue to study it, and where implications of ongoing research on this organism may lead. So, starting with the submarines. Submarines deploy for months-long operations with the highest priority being stealth. Yet these vessels have to stay in constant communication with Central Command. So for years, That entailed positioning close to the surface at predetermined times to float an antenna to send and receive coded messages. Well, that process created a vulnerability of having the sub's location detected. So to solve that problem, the U.S. Navy developed a concept called ELF. And ELF is an acronym for extremely low frequency radio signals. And in 1983, construction of the ELF project began in Republic, Michigan, in the Escanaba State Forest, and became operational 6 years later. So rather than transmitting through the air radio signals, ELF radio signals are delivered by an array of underground cables. Extremely low frequency radio waves are exceptionally long and therefore require a massive antenna. ELF consists of 158 miles of buried cable connected to a transmitter at K.I. Sawyer Air Force Base near Marquette. What made the Republic site ideal was its geology. There, the Precambrian rock shield, the world's oldest rock, is close to the surface. The rock carries the low-frequency signals deep into the Earth's crust, which acts as a giant antenna. So before this ambitious project was approved, extensive environmental impact studies were conducted. A team of researchers led by Johann Brunn, PhD, who's a plant pathologist at the University of Missouri, studied the subsurface microflora at the proposed site. One of the specimens they uncovered was the fungus Armillaria gallica. Now, Armillaria gallica is widely distributed throughout North America, so to have it growing there was not a surprise. The surprise was its size. Through DNA testing, it was determined that this was not a widespread colony spread across 180 acres, more than 50 times the size of Bronson Park. 50 times the size of Bronson Park, but one individual organism. Its mass was estimated to be more than 440 tons, and at the time, it was the largest living organism on the planet. And it's not just the size of this fungus that is remarkable. Its longevity, calculated to be more than 2,500 years, made it both Earth's largest and oldest living organism. So let's refresh a little bit that what are fungi? Fungi were once classified as plants, and they have a kingdom of their own now, fungi. Fungi include unicellular organisms, yeasts and molds, and multicellular organisms, mushrooms. So unlike plants, fungi are not capable of photosynthesis, but rather decompose organic matter or are parasitic by secreting enzymes into a substrate and absorbing nutrients. As for once being considered plants, fungi are actually biologically more akin to animals. And I'll have more on that fact later. The base structure of fungi is hypha, which is filamentous cellular tissue smaller in diameter than a human hair. Strands of hyphae with pliable chitin as a cell membrane form branching networks called mycelium. Mycelium spread below ground or within decaying organic material to find nutrients. Hyphae also carry out sexual and asexual reproduction. Sexual reproduction occurs when two compatible mycelia fuse. This new mycelium is capable of forming an above-ground structure consisting of a mass of hyphae called the fruiting body, a mushroom. The fruiting body produces spores, and as spores scatter by wind or other means, when they find a suitable environment, they germinate. The fungi we're most familiar with, the mushrooms that pop up in spring and fall, again, are fruiting bodies which produce spores. Mushrooms, volume-wise, represent a tiny fraction of the fungus living below ground. This is definitely the case with Armillaria gallica. In the fall, an edible woodland mushroom appears in dense clumps called the honey mushroom, the fruiting body of gallica. Different species of mushrooms are typically distinguished from one another based upon the characteristics of their fruiting bodies, although at times microscopic or even DNA analysis, as in the case of gallica, is required. Fungi fall within three groups as to how they obtain nutrients. One is saprophytic, feeding on dead or decaying organic material, and these are the decomposers. Two, symbiotic, a mutually dependent and beneficial relationship with algae, lichens. And three, parasitic, absorbing nutrients from a living host. Armillaria gallica is both saprophytic and parasitic. Armillaria root disease, which affects trees, colonizes root systems and through what is called necrotrophy, parasitically kills the live root and then saprophytically absorbs the dead tissue for nutrients. It's often referred to as white rotter found on roots. So we know that this Armillaria gallica is huge and extremely old. Another characteristic that sets it apart is that it is also bioluminescent. Aristotle in 382 BC first described the phenomenon known as cold fire on moist or damp wood, and later colloquially named foxfire. Bioluminescence is observed when living organisms biochemically emit a glowing light. So we're familiar with bioluminescence in such creatures as plankton and fireflies. However, bioluminescence is rare in fungi. Only 75 species out of thousands, if not millions, have been identified. Bioluminescence is produced by a compound called luciferin. Luciferin, oxidized by an enzyme, releases energy in the form of a bluish-green light. However, luciferin in fungi is unlike luciferin in animals. Fireflies flash to attract mates. Marine animals do likewise. The question becomes, what advantage does bioluminescence provide fungi? There are a few hypotheses. First, bioluminescence may attract nocturnal insects and other animals, which in turn carry and disperse the fungi spores. A scientist, Cassius Stavani of Sao Paulo University in Brazil, ran an experiment to test that hypothesis. He and his team constructed plastic fungi models that emitted a blue-green LED light replicating bioluminescent mushrooms, which they placed on the forest floor. He found an increase in insects were attracted. Savani states, it appears that fungi make light so that they are noticed by insects who can help the fungus colonize new habitats. Second, while some suggest that bioluminescence is a byproduct of metabolism, in fungi it is a nocturnal function. This is probably true of virtually all bioluminescent organisms, since the light intensity is so weak it cannot be seen in daylight. And as Stavani further states, the circadian control of bioluminescence makes the process more efficient. A third hypothesis is that bioluminescence helps deter predation, but that yet remains to be proven. Fourth, fungal bioluminescence may be a genetic carryover from the early formation of life. A distant variant of luciferin may have limited free oxygen, which was toxic to earlier forms of life before the arrival of microscopic photosynthetic marine organisms, which began a massive production of oxygen. So while all of these characteristics are interesting, one aspect of Gallica is undoubtedly the most intriguing, related to its age. As mentioned, this specimen which is known as C1, so I'll refer to C1 from time to time. But this specimen, known as C1, is estimated to be more than 2,500 years old. By estimated, researchers took two knowns, its size and plausible growth rate, and calculated its age. However, the scientists working closely with this specimen agree that its age may be vastly underestimated. This fungus may have propagated soon after the last ice age, more than 10,000 years ago, once host vegetation was established. What caught the attention of scientists is that over the 2,500 years of cell division, mitosis, it should accumulate an expected level of DNA mutation. Armillaria gallica has an extremely low level of mutation. As stated in a New York Times article that was January 8, 2019 by Joanna Klein, it reads, "You expect to see mutations emerge in copies that are passed on from one generation to the next, but this old beast harbored only about 160 mutations, orders of magnitude lower than expected." So this discovery raised profound questions. While research is underway to more accurately establish the mutation level, the extremely low mutation rate may have implications for cancer research. Cancer's growth rate and mutation is exceedingly high. Further research may identify the mechanism or mechanisms for reducing mutation and apply it to cancer treatment. Klein's article also quoted Tom Burns, a fungal ecologist at the University of California, Berkeley, and I'll read this quote. "The infecting tips of the fungi's rhizomes," and that was, I think that's an error. I think that error was corrected by an editor at the New York Times, because it really should be rhizomorphs, and we'll talk about that in a minute. But the infecting tips of the rhizomorphs could have low rates of cell division, or the fungus could be really good at repairing damage inside its cells, passing healthy traits on to the next generation. Even more bizarrely, the cells may be selective about which copies of DNA they send on to the next generation. Maybe it's a combination of these factors or something else entirely. Then Burns added, "What can it tell us about cancer, which seems to be on the opposite end of the genetic stability spectrum?" As I mentioned, and I saw a few eyebrows go up, it did for me as well, but as I mentioned, fungi are more closely related to animals than plants. Animals and fungi are thought to have evolved from a common ancestral protist, a single-cell organism. And Mitchell L. Sojin, PhD of the Center for Molecular Evolution at the Marine Biological Laboratory in Woods Hole, Massachusetts, says, I'll quote, "The evolutionary affinity between animals and fungi could explain why fungal diseases in humans are so difficult to treat. A lot of the metabolism is so similar that you cannot target a fungus sufficiently without gravely affecting the human host as well." So one could assume that this biological relationship could work for as well as against various treatments. For example, research has demonstrated that turkey tail mushrooms, and I won't give the Latin name on that, may have anti-tumor properties thought to be related to its immune boosting effect. One test tube study found that PSK, the polysaccharide peptide found in turkey tail mushrooms, inhibited the growth and spread of human colon cancer cells. I spoke with Dr. Sojin in June of 2019, and as an evolutionary biologist, Sojin works on the ancestral tree of all living things. He confirmed that consensus is really building that fungi, once labeled colorless plants, are indeed evolutionarily more closely related to animals. He also felt that understanding the reason for the low level of mutation in A. gallica will take some time to uncover, since there are so many confounding variables, including environmental, which must be considered. So whether fungal metabolic processes become applicable in the development of cancer research at this time is still speculative, but there's some potential for it there. A year ago, in October of 2019, I joined Dr. Bruin at the Armillaria gallica site near Crystal Falls, Michigan, along with his two research partners, James B. Anderson, PhD, biology professor emeritus with the University of Toronto, and Myron L. Smith, PhD, Department of Biology and School of Mathematics and Statistics at Carleton University in Ottawa, Canada. In addition to the research team, two others visited the site. Pratima Devkota, PhD, is a Michigan State University professor, and she's researching armillaria root disease infestation of cherry trees in the Traverse City area. And Michigan is the world's largest producer of tart cherries, so that's an important business to protect. She collected wild cherry saplings growing in C1's domain to look for resistant strains of cherry trees. Simeon Wright was the other individual, and he is a forest health specialist with Michigan's Department of Natural Resources, and he was there to survey the site. The group's goal was to gather samples for genetic studies. Anderson specializes in research on population genetics and fungi evolution. Smith, too, does genetic research and the application of genetic markers in life history studies. Shortly after Bruin's discovery, he sought the assistance of Anderson and Smith for their expertise in DNA analysis and work, and they've collaborated ever since. They completed a whole genome sequence for this fungus, C1, and isolated DNA mutations. As part of the initial site research, a section 40 meters by 120 meters of hardwood forest was cleared, and red pine seedlings were planted and monitored for several years to study the primary specimen of A. gallica C1 and to identify and map other individuals of the same species as well as other Armillaria species. So it's easy to assume that A. gallica is this massive vegetative body beneath the forest floor, sitting passively and docile for centuries. The biology of Armillaria is anything but passive. It forages for its nutrition. A distinguishing characteristic of Gallica, I should really say Armillaria, is a modified hyphal appendage called rhizomorphic mycelium, or rhizomorph, rhizo being root, morph, like or form, which has a protective sheath. Another common name for A. gallica is shoestring root rot because of its tough, sinewy, dark brown threads penetrating the soil and host tissue. Rhizomorphs perform a variety of functions. As they spread through soil, they protect the mycelium within. Rhizomorphs also produce an assortment of chemical compounds which ward off microbial assaults, repulse host plant defenses, and combat rival fungi. Once a potential host is encountered, then it penetrates the tissue. If a potential host is healthy and vigorous, Gallica may establish itself on the root system and wait for such time when environmental conditions or insects and disease stress the host and reduce its resistance to Gallica's parasitic invasion. Once Gallica's hyphae penetrate plant tissue, it deploys some highly advanced adaptations. One is the formation of a flat mat of mycelium known as a mycelial fan. And I could show you an image of that later, not of Gallica, but just a mycelial fan. This fan forms a protective layer to defend against the tree's response or competitors and allows spreading hyphae to release specialized enzymes to digest plant tissue, lignin, and cellulose. And lignin and cellulose are tough substances. So that's some pretty powerful enzymes that get that work done. A second adaptation is the formation of a distinctive barrier, dark in color due to melanin, called a pseudosclerotial plate. This plate, which woodworkers admire, is spalting. Those are those striking color patterns in wood. You know, when they turn bowls and that, they love that. That plate partitions plant tissue to optimize Gallica's nutrient acquisition. During the October visit, or when I was up there last October, Anderson, Smith, and Brun located a fallen tree and examined below the bark layer. They found a rhizomorph running its length. They systematically collected sections of the rhizomorph and noted each sample's location. Once they confirmed the rhizomorph is Gallica C1, DNA analysis to identify genetic mutations will be conducted. And Anderson says that the samples we collected that day, represent years of research. As a matter of fact, I'll just throw in a little note here. I didn't put it in. When I was talking to him about it, is that part of the research they do and they accumulate it isn't so much for their research, and they're fascinated, they're engaged in it, but they know that this research is going to have to be picked up by others, down the road. So it's a long-range process that they're looking at. The team also set up 2 field experiments to study the foraging behavior of Gallica. For each, they cut 100 30-centimeter sections of poplar saplings and drove them into the ground about 20 centimeters deep in an evenly spaced 10 by 10 stake grid. All right, they call this technique baiting. And one site surrounded the tree, the fallen tree from which the rhizomorphs were collected. They were to return, and I don't know if they did this year, but their intent was to return in 2020, probably about this time of year. But they would go back and examine those stakes to identify rhizomorph incursions into those saplings. So I have a little bit of a video. Kind of introduce you to the name of the real people and get a chance to see a little bit of this. This is 54 minutes, so no, it's not. There we go. In October of 2019, I met up with a research team at the Armillaria site in the Upper Peninsula. This team of researchers has been working on this fungus for about 30 years now, and they were up to collect specimens for continued DNA research. And here they're collecting rhizomorph samples from the site location. They found a log where there was a rhizome about the entire length of that log, so that was ideal for collecting at specific intervals along the log so that they noted the precise location, and from that then they could determine, as they look for mutations, where in the growth cycle that those may have occurred. In addition, they set up some test sites for the following year where they cut poplar saplings and drove them into the ground in a very precise grid. Again, all that detail that was recorded, 100 stakes in 2 different sites that they call baiting. And they'll come back next year and then check these stakes to see if there's been any contact with the rhizomes from that fungus, and that will help determine and continue the research on Armillaria gallica's foraging behaviors. The next morning, they took the samples that they collected to prepare them for the trip back to the labs in Canada. And here they have a rhizomorph, cutting it into almost uniform little sections where they will then
place them in a bleach solution for about 10 minutes or so to kill any bacteria or other microbes that are on the surface so that it doesn't interfere with the DNA collection. And once they were blotted dry, they put them in petri dishes with a agar base and then sealed them up, ready to take back to the labs, as I mentioned, and along with all the necessary paperwork and permits to carry such material across the borders.
Yeah, when we came back from the field that afternoon, they pulled into a store. I just hit sleep. I thought, hey, these might be just regular guys stopping by and picking up some beer to take back to the hotel. They came out with a jug of Clorox. And they just used Clorox bleach for the process. So they were true blue, and they weren't monkeying around at all unless it was dinner time. So anyhow, the story of Armillaria gallica is engrossing and no doubt has more to unfold. Acclaim for this humongous fungus was not confined to the scientific world. In 1992, which Bruns describes as a media crisis, the first scientific paper about the discovery of this organism was published in the Royal Society Proceedings B. That article was picked up by the New York Times, which ran a front page story headlined, "World's Largest Organism." In turn, it was carried by network news programs and the media at large. And Johnny Carson and David Letterman even brought it up in their monologues, right? Run said the phones immediately started to ring from people around the world, and this humongous fungus story took off. And not only did the story take off, but the story also generated misconceptions and exaggerations. It was reported that this humongous fungus was a giant, throbbing, malodorous creature alive in Michigan's Upper Peninsula. And A. gallica quickly became very popular, and reporters and people wanted to visit the site. News of the story caught the attention of Harvard paleontologist, evolutionary biologist, and popular science author Stephen Jay Gould, and Gould published an essay in Natural History magazine in July of 1992 in which he commented on the sudden celebrity of this humongous fungus. The small town of Crystal Falls was inundated with curiosity seekers. The owners of the town's Ben Franklin store, and it was one of the original stores in the chain and still in business today after 90 years, Jeff and Sturia Surgeon, and I met them, wonderful people. They recognized the public relations value of this newfound fame for their community, and they established an annual event each August called the Humongous Fungus Festival in collaboration with the Iron County Economic Chamber Alliance. So if you want to go up and get a t-shirt that says Humongous Fungus, well, they've got them. So the discovery and research of Armillaria gallica and this particular specimen has spawned many fascinating aspects for continued study. Its significance centers on the fact that as one question is answered, especially in science, it generates further questions. Over dinner, Bruin, Anderson, Smith, and I had a thought-provoking discussion on that phenomenon, which they confirmed is a prime challenge in the pursuit of knowledge. It may indeed turn out that this humongous fungus leads to breakthroughs in cancer, DNA research, and understanding of specialized enzymes which have evolved over eons may lead to tangential agricultural applications. And likewise, research on host defenses to protect against armillaria disease may initiate additional lines of inquiry. The good news is this humongous fungus is much more than a curiosity. It's set into motion a momentous scientific quest, the results of which may far exceed its size and longevity. That's our story. But I maybe if I could mention a couple other things, and then we can take some questions. one just happened to be on Saturday. I'll flip to that the one I was going to do first next. But on Saturday, I went to the farmer's market, and there's a table where they sell mushrooms. And this nice gal, young gal, I started talking to her, and I said, hey, you ever do anything with honey mushroom? She knew. So she was talking about the honey mushroom, and I was getting this conversation going with her. And then something I had not known at all, she said, oh, there's also a shrimp of the woods. You've heard of chicken of the woods fungus, but there's one called shrimp of the woods. And shrimp of the woods is Entoloma abortivum, abortivum is the scientific name. That mushroom, it was thought that armillaria was parasitic on that mushroom, but it's the other way around. So armillaria has a parasitic fungus that attacks it. Now, I got home and I thought, I'm going to look into this a little bit more. That's kind of interesting. I didn't even know about it. Well, then come to find out, and she had mentioned this. She said, yeah, you pick the one that's of that mushroom, you pick the one that's on the armillaria, on the honey mushroom, I should say, on the honey mushroom. And sure enough, as I was looking into this, that they do not recommend that you collect or eat the samples of that species of mushroom collected on its own. The one that's safe to eat is the one that has parasitized the Gallica. Now, what on earth would have transpired there? And they look entirely different. So the other that I wanted to bring up, and I didn't want to work this in because I didn't want to go off on a tangent when I was giving a talk, is that this phenomenon of, to me, I don't know if it's a phenomenon, that's probably not the right word, that fungi are more closely related to animals than plants. There's a fungus called Ophiocordyceps unilateralis, and that is in the tropics. And what this fungus does is it affects ant colonies, and ants that live in the canopy of these tropical forests. When they're infected, it changes their behavior. The ants go down to the ground level where the temperature is better and the moisture levels are better. And then the ants crawl, they find a stem and they crawl and they attach with their mandibles onto the back of a leaf. And there they die. And then the fungus sprouts and the spores are released. But think of that, a fungus that actually takes control of behavior and body movement, which is amazing. And then the other, and I can send you this if you'd like, because it's too much to cover today. That should decimate any ant population totally, as that would spread. But there's a fungus that attacks that fungus that keeps it in check, and lo and behold, as they start researching that relationship, they're finding antibacterial and other agents that are produced by those fungi. And then the last that I'd like to mention, when I said that there were three main divisions as to how fungi are categorized by the way they obtain nutrients, and I'll show you this. That's the textbook explanation. How many of you know Dr. Paul Alexia with K College? Terrific fella. And he's a mycologist. And I had talked to, last year I talked to Paul. I've known him for a long time. Just want to make sure I had some of my facts straight about it, explaining what fungi are and not. But as we were talking, Alexia told me about, I hope I can blow this up. that coming up? Okay. That hyphae also, not every fungus, certain species, no doubt, hyphae can form trapping rings. And that worm-like creature, that's microscopic, that's a nematode. That when nematodes are traveling, if they pass through that ring constricts and holds that nematode, and they can feed off that nematode. So I think that the textbooks may also have to now include carnivorous. But to form those kind of amazing structures, isn't that incredible? So anyhow, that's my spiel, and I'll be happy to answer any questions if I can. Yes, Michael. That I don't know. I don't have that information. But these fellows that are working with it certainly would have that.
So the mutation has to do with the size of the genome, right?
Yes.
The bigger the genome, the higher the rate of mutation. But in human and most of the animals, the DNA polymerase, which is in charge of manufacturing of new DNA,
Yes.
has a self-correcting system which is called proofreader. So as the new nucleotide adds to the chain, if that nucleotide was wrong, let's say instead of adenine supposedly added thymine, immediately we correct that. So as the chain progresses, you have a DNA which is very much like the mother DNA. So the rate of mutation in human, at least human genome, is 1/7 million nucleotide. This new virus corona that we have also has this self-correcting system in its replicating polymerase. For that reason, the mutation of coronavirus is much lower for the size of the genome. It has 29,000 nucleotides. For that size, to have very, very few mutation is amazing. That has to do with that system of self-correcting. I wonder if this fungus most likely has similar system of self-correcting itself, because over so many years, the DNA of this fungus hasn't changed,
Yes.
as you said.
That could be.
That's amazing.
Yeah.
And also the other theory is that because it's under the ground, it's out of reach of ultraviolet and cosmic radiation,
Yes.
which is constantly bombarding the planet. So then the rate of mutation would be much lower for those creatures that live underground. And the farther they are, the less they are at risk, because the only thing that can go through would be neutrino, which has no effect on the living cells. So that is, and also I found out that this mushroom you're talking about, also like this, maybe bigger, is present in Oregon.
Yes.
So it's
It's
not
larger in Oregon.
Exactly. It's not just the
Yes.
this part of the United States. So that's my comment.
Yes. And with the with that genome, they've given me some of the scientific papers. So I'm sure that's in there somewhere. But what caught my attention, there's two things. One is that they had an expectation for what that level of DNA mutation would be, and this was orders of magnitude lower than what they had projected. So they may have done the same kind of math that you did.
No, they figured out the biology. They figured out in the technobiology. We know
Yeah.
exactly what is the rate of mutation. So that's
Yes.
how you can also read the age of the organism.
Yes.
When you go back, based on the number of mutations, you can age them. You can say that we came from monkeys, because that's based on the SNPs, changes in the structure of the chain of DNA. So because the rate is mathematically very fixed.
Yes.
So if I know that many have mutated, I can multiply by rate and then find out how many years ago was this creature on the planet.
And when I talked to Dr. Sojin, the fellow from Woods Hole, when we were talking about what would be the possible mechanisms, and he talked about these, we talked about these variables. environmental was there, and we discussed that being underground and in a cooler environment and less, sunlight and all those kinds of things would all have to be taken into consideration. That, you know, uh, yes, yes,
That was discovered up north.
uh-huh.
You said it was one organism.
One organism.
Does it have one nucleus? And
It's there.
I mean,
Each of the each of the cells would have a nucleus in it, but they do the DNA testing on this, or the analysis, and it's the very same genetic material wherever they happen to sample it. I mean, it's absolutely precise that it could only be that one individual. And when they say they estimated 440 tons, as I talked to these fellows as well, as they were saying, they tried a couple of methods. One, they just dug a square plot of soil, a foot by a foot by a foot, and went in to see if they could kind of get an idea of how much fungus material was there. That's really, really crude, and it just happened to be one spot. But they also realized it'd be almost impossible to measure the mass or the volume of the mycelium that's in trees or in branches or way down low. That's why that may have thrown off that age calculation. But the best they could come up with that would be reasonable was that this is 440 tons and over more than 2,500 years old. Yes, Barbara. It forages for food, or nutrition, I should say, not necessarily food. And as I was talking to Brun and the other fellows up there, when they map this, they do this sampling all over. It's in an area about 50 times the size of Bronson Park, this organism. But what they also see, and why they're looking at these studies for this foraging behavior, is that it doesn't waste its energy if there's not a lot of potential nutrients there. It will move and wander, and it probably actually follows different opportunities as vegetation grows or doesn't grow. So it'll move to optimize its nutrition. Yeah, in a short season. Yeah, in a short season.
It has to be close to the roots of the trees,
Yes.
because that's how you get its nutrition, right?
Yes.
Otherwise, it doesn't serve any purpose to go deeper.
Yes.
It has to be where the food is, the roots.
Yeah.
So I would say close to the surface.
Ross?
Sounds to me like you're describing the only immortal that we discovered on Earth.
Yeah. Well, the people at Crystal Falls hope it keeps going. That's for sure. Dude? Oh, I would imagine it varies, but they say that that's the place on the planet. It's one of the places on the planet. It's the closest to the surface. So I don't think it would be, maybe more than 6, 8, 10 feet at most in many places. But they were able to bury that cable, so they'd have to be able to get down a little bit to be able to do that. It's the oldest, yes, yeah. And I also, too, I should say, and I want to thank Dr. Brun for this, and I was shocked, is that as I started looking into this, I found the name of the guy that discovered this, and I never know if they're still alive or around or whatever, but anyhow, I kind of tracked it down and found a way to get a hold of him. And as I was talking to him, just an interesting fellow, he said, look, rather than doing this stuff and email and over the phone, why don't you just meet us up there when we go back up in October? I was thrilled to have that invitation to go up and be on site with them and meet them and talking to them personally. So, terrific people and willing to share. Howard?
Just a quick question: how fast is this thing growing in terms of expanding? And the other part of this would be, is there any point of origin? Has that been identified, that kind of the nativity of this organism, and has it spread out, and is there a rate or predictable growth? Will it swallow the whole UP eventually?
There are two things. One, that did come up in conversations, that there was some thought that I wonder if we could find that Genesis site, to be able to find that. But they weren't able to really substantiate anything close where they'd be able to say, this is where it started. But as far as the growth, as long as the environmental conditions and the nutrient sources are there, it can. There's out in, I think it's North or South Dakota, there's a place called the Pando. Have you ever heard of that? It's aspen. Actually, that may even surpass the armillaria, because here's this beautiful forest of aspen, just growing, and landscape photos. It's one individual because it spreads by runners, and its age is far older than that. But that was a different process. So that's kind of interesting as well.
Jim, thank you very much.
All right.
We appreciate
So
that.
eat your mushrooms.
Oh,
Yeah,
okay.
wasn't that interesting? That's talking to that gal Saturday, at the farmer's market.
Yeah.
And my jaw dropped when she starts filling me in on this other aspect. So, like I say, when one question is answered, it just leads us to another one.
I hope you can all be here next month. We have a really great presentation on solar power and solar energy, solar panels, and everything else by my friend Sam Fields. But we also have something special here, Bob, is we have a birthday baby here. And we will celebrate your birthday by not singing Happy Birthday, Ross Stancati.
Hey!
No age.